Assalamu alaikum and very good morning to everybody. We have been joined by our speaker over here. Sir, am I audible, sir?
Yes, I can hear you.
Oh,
Yes,
thank you, sir. Most welcome to this meeting, sir.
thank you very much for inviting
You
me.
have spared your time and you are with us this morning, and we are really blessed to have you this morning with us. So, before I formally invite you for the lecture, please let me introduce yourself to my participants. My dear participants, this is with us this morning, Dr. Arvind Kumar. He is the Director, International Research, Rice Research Institute, South Asia Regional Center. And he prior this moving to ISARC, Dr. Kumar worked as interim head rice breeding platform at IRRI. Dr. Kumar also led the rain-fed lowland South Asia plant breeding group. Then he did his BSc in 1989 from Indira Gandhi Rishi Vishwa Vidyalaya with first position, and then MSc in 92 again from the same institute, again with a position. Then his PhD from the same institute, International Center for Genetic Engineering and Biotechnology, New Delhi, again with the first position. So you can understand his academic standing there. And then he has expertise in trait development for drought, aerobic rice, and dry direct seeding rice. He has developed 61 rice varieties. Those are released in 10 different countries, published 146 peer-reviewed manuscripts, and more than 30 book chapters. Dr. Kumar identified 7 genes for rice gall midge resistance and several QTLs for grain yield under drought, for grain yield under dry direct seed situation, for traits important to aerobic and dry direct seeded rice like early vigor, and then lodging resistance root traits related to high nutrient uptake under dry direct seed situation, and nematode tolerance, and 2 stable QTLs each for high iron and high zinc in rice. Dr. Kumar has supervised 33 scholars and postdoctoral fellows. This a brief about Dr. Arvind Kumar, and we know his position is presently the director IRRI, and it's our the pleasure that we have a person with this profile and dignity with us this morning. And I hope all my participants will enjoy his lecture and his wisdom, particularly in the rice. And we too in the Valley of Kashmir, we cherish rice twice, thrice a day. And then that will be as a bonus to our agriculture scientists. Dr. Kumar, we have with us this morning the participants from agriculture universities throughout the country. It's not from the Kashmir only, throughout the India. We have agriculture scientists, the junior scientists, senior scientists, professors with us this morning. And they are from agriculture and allied fields, purely agriculture, as well as from animal sciences also. So you are most welcome here, sir. You can take over now for your presentation, and you can share your slides also. Uh, then,
Yeah,
yes, Kumar, over to you, sir. You can put
thank
your
you.
video on, please.
Yeah, just a second. Just
Sure,
a
sure,
second.
sir, sure, sir.
Okay, thank you very much for the introduction. I hope I am audible to everybody.
Yes, sir, yes, sir.
And can you see my screen? No?
You share your screen, please.
Can you
Yes,
see?
yes, it is now coming. Yes, exactly.
And it's let me make it full. Okay.
Yes, yes, yes. You put your video also on.
Yeah, let me do it. How to do it? Okay. Sorry. Yes.
Yes, yes, yes.
Oh,
Yeah,
yeah. It's okay.
yeah, yeah.
Everything perfect.
Yes, you can just make it a presentation view. Oh, yes, yes. Click there. Yeah, good, good.
Okay. Thank you very much for inviting me to this important initiative in between COVID-19. And to be very frank, it just got out of mind just before I got the call. I said, "Oh, today is." I had some idea 27, so I said, "Oh, it is today." So thank you very much for reminding me well before time, so that I could quickly get connected to all of you. And thanks to those around 50 plus people who have joined to this talk. I try my best to make this talk as interesting as possible. So we will be talking about breeding, and we will be talking about breeding, that how breeding has progressed for the abiotic stress tolerance and mostly for the drought tolerance. And as you know that drought is a complex trait, you know, and we will go through that what we have done at ERI, and also how closely we have worked with some of the partners in India for drought tolerance. You can, uh, if you want, you can stop me in between. You can ask me some questions, or if you wish, we can have the question answer in the last.
Sir,
So
we'll come
let
into the last.
okay. So we will have in the last.
Yes.
And so let's move ahead with this presentation. Genomic assisted breeding. We will talk mostly about the drought tolerance. So the outline of this talk is major QTLs for granule under drought that that we have identified. So we will uh, bring some story that how we identified those QTLs and how those QTLs are different from others. Then we will talk about undesirable linkages. When you work on the traditional lines and traditional donors, then you do see some undesirable linkages coming which affect the other traits for which we are breeding. Maybe it is the high yield potential or it is the grain quality. And then we will talk about the marker assisted breeding that we have done with identified QTLs. Then also marker assisted selection of QTLs with genes for other traits. And then we will, in the last, we will talk about the superior haplotypes and their use in the breeding. Now, the whole aim of this talk is that to encourage, I don't know how many of the participants are plant breeder, but irrespective of what you are, you know, the aim and the story is that as researchers, we have to keep ourselves updated with the new knowledge and the new technique if we really want to be competitive in this knowledge changing world. And that is very necessary. So this story is more aligned to plant breeding, but I think that holds good for anything, that holds good for pathology, that holds good for entomology, that new techniques and technologies and new knowledge are coming, and which as scientists we need to keep ourselves updated, and we need to keep ourselves ready to use such technologies and techniques to be able to stay competitive in this world. So I started a journey, I would say 2004, working on drought. And I have no hitch in saying that at that point of time, I didn't know that what I will work on drought. So when you say drought, you have so many things. People talk of the leaf drying, leaf rolling, transpiration, osmotic adjustment, you know, and I thought whether we will be able to increase the yield by improving these traits. That we will be able to increase the yield by using these traits, but there were reports also that that there was no increase in yield by using these traits. So I said, what we should do. So I was working with my supervisor, and we decided that we will work and try to improve the grain yield per se, without worrying initially that from where this increase in yield is coming, whether this is coming from improvement in the leaf rolling, where there is a less leaf rolling, or whether this is coming from improvement in transpiration efficiency, or this is coming from osmotic adjustment, or this is coming from the better root system. So we decided to prove that the selection for grain yield, which works very effectively in the irrigated ecosystem, because we develop our variety based on the direct selection that we do in the field for the improved yield, whether that will work in the drought also. And how we can prove it? We can prove it by proving that under drought, yield has the same heritability which determines the selection efficiency in the large way as in the irrigated ecosystem. And we did number of experiments at in fact some at Raipur and some at Iri, and you can see that the heritability for the grain yield under drought was similar to that under irrigated situation, which I have put in here non-stress situation. And therefore we were able to prove that the selection for yield under drought is going to be as effective as under irrigated ecosystem. The second question was whether we will be able to develop variety which has the high yield potential and good yield under drought. And then we also proved that the correlation between yield under drought situation and the yield under uh, the normal situation, which is irrigated situation, is less low but positive. So that means that if we deal with the larger population, we will be able to capture that positive correlation. So having done these experiments for around 3 years, we decided to move ahead after this, that we will identify QTLs for grain yield as such under drought. And then once we have the QTLs, then we will try to know that whether this increase in yield is coming from osmotic adjustment, transpiration efficiency, or root related traits, or any other traits. So we moved ahead with that we will identify QTLs for drought tolerance for grain yield. So, but we were not the first one to do this. There were many QTLs reported, and in fact, if you search the literature for the QTLs, you will see hundreds of QTLs for most of the traits on which people work. But most of these QTLs were not in the background of high yielding variety. So the efforts were made by the earlier researchers that they identify the QTL in the background of the most susceptible variety. And so when you try to use such, because you want to show that you have identified a QTL that has large effect, and so you use a weak background which is highly susceptible. But when you try to use these QTLs to improve prominent varieties, you don't get that effect. So we said we will have QTLs, we will identify QTLs in the high yielding background. So we selected number of varieties: IR64, Bandhana, Swarna, Sama Masuri, MTU 1010 from India, then Savitri from Nepal, TDK1, sub-1 from Laos, and so on. BR11 from Bangladesh, Saro 5 from Africa, so many varieties. We were also knowing that there's a lot of QTL into genetic background interaction, which is a constraint, and so we have to be able to prove that the QTL that we identify is showing effect in multiple backgrounds. So it's just not that you identify a QTL in one background and then become so confident that this will work in the next background. Not necessary. So we took a strategy where we identify a QTL in one background, but we prove its effect in the two other backgrounds, and in some cases we identified the QTLs in three backgrounds, and then we could say that that these QTLs are effective. And then we also saw, and you will see as I go through my presentation, that the effect of the individual QTL was between 300 to 500 kg per hectare. So they were able to improve the yield under drought by 300 kg per hectare or 500 kg per hectare in between these, which probably is not good for farmer, that if you just give him a advantage of 300 kg per hectare, then he will use your variety. So we decided that we will pyramid 2 to 3 QTLs to get 1 ton or higher yield advantage per hectare under drought, so that our varieties that we breed are utilized by farmers. So with this background, we started the QTL identification program, and the table that you see here is a compiled research of over 10 years, between 2004 to 2014 or 15. And so you see here that every QTL that we identified starts with QTL 1.1, then this is on chromosome 1, which is near the SD1 locus, very closely located. And you will hear something about the story that how this QTL was tightly linked to the SD1 locus. And then there are QTLs on chromosome 2, then on chromosome 3, then on chromosome 4, then chromosome 6, then chromosome 8, then chromosome 9, chromosome 10, chromosome 11, chromosome 12. And many of these QTLs, those that you see in the red, they have shown effect under both lowland and upland ecosystem. So you can see here that for example, QTL 1.1 have shown effect under both, QTL 2.2 have shown effect under both, QTL 2.3 under both, QTL 3.2 under both, QTL 6.1 under both, QTL 12.1 under both upland and lowland ecosystem. So evidently, and this was not heard of, that a QTL can work in both the environment, and so and we were also surprised that in the upland where you go for the direct seeding, these QTLs are working, and in the lowland you go for the transplanting, then these QTLs are working. And we did show the effect of these QTLs in the different background. So for example, 1.1 coming from so many donors, and you see how many donors we use. We use N22, we use Dagardesi, we use Apo, we use CT9993, then Kali Os, then Basmati 334, and we proved it in three background and three high-yielding varieties from India, Swarna IR64 and MQ 1010. And so is the story. So we got confident that these QTLs are working. These QTLs are major effect QTLs, and when we presented this story one by one, these QTLs were coming. The first one to be identified was the QTL 12.1. So I still remember that when I presented this QTL to the donor who funded this story, and they were like, oh, is this real? And then they gave us the challenge, and in the terms I was benefited that challenge was accompanied with a project. That okay, we give you money, you progress this QTL into a variety, and then prove us the effect. So this. So QTL identification, we have, I would say, around 6 QTLs as of now, which we are extensively using in the breeding program. And these QTLs are being used in the breeding program for drought tolerance in many institutes in India also. And you will hear something about this. But then the second part of this story begins, that whether these QTLs show the effect. But before we go there, let's talk about the drought tolerance and the germplasm from where we use the drought tolerance. For example, I told you N22, Dagar Desi, Apo, CT993, Kalios, Basmati 334, Adaisel, any of these, Morobrakan, any of these lines, that if you know any of these lines, you will feel that what is my next statement is correct. Drought tolerant germplasm are mostly tall in height. And so most of the drought tolerant germplasm, they are tall in height. Drought tolerant germplasm mostly possess low yield potential. Their yield potential is very low. If their yield potential would not have been low, they would have been used under cultivation. So they are not in cultivation. Drought tolerant germplasm are very early in duration. Most of the germplasm are very early in duration. And I think one of the traits that they have is that they try to escape also the drought. Drought tolerant genotypes have poor grain quality. So if you have seen N22, it's totally different from basmati, or totally different from any variety that we eat. Drought tolerant genotype germplasm have efficient root system, and that is why one of the reasons why they are tolerant to drought. Let us remember this. These 5 or 6 statements, 5 statements will be very useful in the later part of the presentation. So what we did was, after we identified the QTLs, we took 129 diverse lines, and these diverse lines included some of the drought tolerant donors that we have talked, but this also included the early green revolution varieties. For example, IR8 is there, which is a early green revolution variety. Then we have the mid green revolution varieties, which includes Jaya, which includes BR1, and then the late green revolution varieties, which included Swarna, MTU 1010. And when we studied this for the alleles that they carry, we did find that they separate. That means they are not grouping in random. Their grouping is totally different. Drought tolerant genotypes are grouped separate, and drought tolerant donors are donors. They go into group in a separate group. Then even the drought tolerant new varieties that we have developed, early duration green revolution varieties, they form a separate group. Mid duration green revolution varieties, they form the separate group, and late duration green revolution varieties, they form a separate group. So what is happening? Sorry. So when we try to see that the drought tolerant alleles in them, we found that that in green revolution varieties, the drought tolerant alleles went on decreasing from nearly 39.57%. If you see the cluster 1, this went on decreasing in the early green revolution variety, which is cluster 2, to 20%, then the mid green revolution variety to 18%, and then the late green revolution variety to around 16%. And this continues. But when we started our drought tolerant breeding program, we were able to increase this up to 27%. And the alleles for the traits for which green revolution varieties were bred, the high yielding, dwarf plant height, this goes on increasing from 33% to 58%. So evidently what happened, that in breeding for the during the Green Revolution era in breeding for high yield and dwarfism, we did lose the alleles for drought tolerance, and therefore the new varieties are more susceptible to drought than the old varieties, traditional varieties that were being cultivated. And this happened because of linkage between the traits, drought tolerant traits, and the traits for which were present in the traditional varieties. And because we wanted to, for example, one trait was the tall plant height. So drought genes, alleles are linked to the tall plant height. So when you go for the dwarf plant height, along with the losing the tall plant height, you also lose the drought tolerant alleles. So what we did was that I told you that Qtl 1.1 is located very near to the SD1 locus on chromosome 1, which determines the plant height, and it was linked to the tall plant height. And for number of years, there was this controversy whether this is the drought tolerance is because of the pleiotropic effect of SD1 or because of the linkage. We developed a large population, recombinant population, when we were trying to find map the Qtl 1.1, and we broke this linkage. And so what you find here is that a plant, we developed plant with the same plant height as that of the Swarna. You can see here Swarna has 84 centimeter plant height, and our drought tolerant lines have the same plant height, but they were more drought tolerant. So we broke the linkage of tall plant height with the drought tolerance. And so now these lines, that 3 lines that you see here, can be used in the breeding program, and they don't carry the tall height allele. And evidently you will see here that the yield of these lines under normal situation is similar to Swarna, and you see a advantage for yield under the stress. If you see the G by E stress, you can see that how much advantage over Swarna is there. The same way, 6.2 was linked to the earliness. We broke this earliness, and you can see we developed the lines with nearly same days to flowering as that of TDK1, same yield potential as that of TDK1, with yield advantage under drought. The same was true for 3.1, which was linked with the low yield potential. And in fact, we broke this linkage and we developed the line which has the similar plant height, similar yield as like Swarna, and the same similar and with yield advantage under drought. And Qtl 2.3, again, this was for the earliness, and we developed here in fact the lines which has the later duration than Bandhana, but still you have the yield advantage. And many of you know that Bandhana is itself a drought tolerant variety. So if you get a yield advantage over Bandhana under drought, that means that there is that those QTLs are really very effective. So having broken this undesirable linkage, these lines, if anyone uses in the breeding program, then you don't get those complications of linkage drag, and your breeding becomes smooth with use of the markers. So we started the journey to prove that these drought QTLs are real. And as I told you earlier that 12.1 was the first QTL that we identified, and this story is very interesting. We made this cross between Bandhana and Vedaram. You can see down in the table that Bandhana and Vedaram, the two in the bold, they were the parents of this cross. And the idea was that Bandhana is tolerant to drought, but Vedaram is susceptible to drought. So the idea was that we will get a QTL from Bandhana, because Bandhana is highly drought tolerant, and that is how we do it. Even for the disease resistance, we take a drought resistant parent, we make a cross with the susceptible parent, and then we try to map the QTL. But to our surprise, this QTL 12.1 came from the susceptible parent Vedaram. And the challenge was that we improved the yield of bandana. So we introgressed this QTL into bandana. It took us 2 years to introgress the QTLs. Then we developed the line. We followed the marker-resisted back cross method. And you can see here that this QTL did improve the yield of the bandana under drought without any compromise on the yield potential. So when you see the upland stress yield, you see that the three lines yielding higher than bandana, and when you see the upland non-stress yield, you see the yield of these three lines are in fact more than the bandana even under non-stress. We send these lines to Hazaribagh for testing, and we did see the effect under the different kind of stress that we used to get. That these QTLs, as I said, I've always given 300 to 500 kg per hectare yield advantage under drought. So this was the first success. The photograph also is evident. It shows very clearly that bandana is tolerant to drought, but it also shows that the new lines which has been developed are having higher tolerance than bandana under drought. So this was our first success. We proved it that QTL 12.1 is real, and the QTLs for drought tolerance that we identified are working. Now, for the second proof of marker-assisted backcross breeding, we identified QTLs in the background of IR64. And you did see that, the IR64 was one of the recipient parent for QTL identification. And many of you know that IR64 at one point of time was mega variety and was grown on somewhere around 10 million hectares land. And it became mega variety just not because of its yield potential, but also because of its resistance to several insect and diseases and the high grain and cooking quality. So we took this challenge, we introgressed 3 QTLs into IR64. And the challenge here was that we need to get a line which just doesn't provide the yield advantage under drought, but it also provides the similar yield under normal situation. Is a dwarf variety, but it has the same cooking quality as that of IR64. So we do the RVA testing, rapid viscous analysis test for the cooking quality, and fortunately we got several lines. At least you can see here 3 lines which has the similar cooking quality as that of IR64. And to maintain this or to achieve this, in fact, we managed a large marker-assisted backcross breeding program, because we fear that that probably many of the lines will not have the same cooking quality as that of IR64. And so we did RVA testing for around 50 lines. Out of the 50 lines, we identified 3 lines. Now, this story is very interesting. This story is interesting that some of these lines, one line is released as variety DRR42 in India. It's a central release, and it's released for, you can see, Jharkhand, Tamil Nadu, Andhra Pradesh, Telangana, Bihar, Chhattisgarh, and Tripura. 1, 2, 3, 4, 5, 6, 7 states. And the news that I have is that this year, this has the highest breeder seed demand. So can you imagine that a drought tolerant variety having the so high breeder demand? But this is happening because marker-assisted backcross breeding allowed us to maintain the cooking quality and the grain quality and also the high yield potential. Now this variety is also released, this line is also released as a variety in Nepal with the name Sukhadhan 4, and also 3 lines are released as variety in Myanmar. So this line IR64 QTL is moving up. So we got more confidence that this is working. And so many of you who have seen the flood and drought in the rain-fed environment know that many of these areas are prone to both. At early season, you can have the flood. At the late season in October and November, you have the drought. And so we said, can we combine flood tolerance with the drought tolerance? And at that point of time, Swarna sub-1, the first flood tolerant variety, is the sub-1 gene, was already released. So we took this line, we introduced 3 QTLs in this line, 1.1, 2.1, 3.1, and we developed several lines. And this table is evident that, the lines with the QTL and the sub-1, uh, in the submergence, they survive. But under drought, you can see that the lines with the QTLs yield higher than the Swarna sub-1. So under submergence, you can see the lines have the same score between 2 to 3, where some of Swarna sub-1 has 4. But under drought, you can see the 1 ton, 0.8 ton to 1 ton yield advantage under drought, and maintaining the similar yield potential as that of Swarna or the Swarna sub-1. Now again, this is just not that we developed a line and we left it. We put it into testing in ECRIP because we do comprehensive breeding. We go for the plant type, we go for the grain type, we go for the high yield potential, we go for every trait that is possible, and we put it into ECRIP in India to see. So there is a variety release CR Dhan 801 in India, and there are 2 varieties, Bahuguni Dhan 1 and Bahuguni Dhan 2, are released in Nepal. So more interesting is that after seeing the IR64 success, people started in India, started using these QTLs into the breeding program. And I put you here the story, 2 story. One is from Hazaribagh that you can see they have used it for IR64, and they have used it to develop Anjali solvent, and you can see the effect in the photographs, and Bandana. And whereas the DRR had used it for Samba Masuri. And in fact, Department of Biotechnology, they started a project which I requested them, and which Dr. N.K. Singh from NRCPB, he led that project with several partners. And in fact, QTL2 variety, I don't know if any one of you were associated. So 4 varieties have been released from that program where these QTLs have been used. So great experience to see that these QTLs are moving into the national breeding program. Now we wanted to move to the 4th stage, where we say that we combine the drought tolerance QTLs with the disease resistance, submergence tolerance, and we bring as many genes as possible together. So as of up to now, you were hearing about the marker assisted by cross breeding. But now you hear about the marker assisted selection. No backcross breeding. We want to develop a new variety that has tolerance to drought, that has BLV resistance, that has Galmi resistance, that has BPH resistance, that has blast resistance, that has submergence tolerance. And so we took a comprehensive crossing program. And you can see here that 1, 2, 3, 4, 5, 6, 7, 8 parents were involved. And it took us, in fact, 2 years, 4 seasons to just attempt the crosses and ensure that we are able to make all the crosses. And you can see the number of F1s as we went into the complex process, the number of F1s that we should be getting, we kept on increasing. So we started with 200 F1s, then we went to 500 F1s, and then we went to the 3rd stage is the 1500 F1s, and then we went to the 4th stage, we went to 3000 F1s. And then we grew a population of 15,000 F2. And then we run all these markers for the grain yield under drought, bacterial blight resistance, BPH resistance, Galmi resistance, blast resistance, submergence tolerance. The idea was that we combine these 15 to 16 genes together. Did we succeed? Not fully, but up to certain extent. So we were able to bring up to 10 genes together. There were some plants where there were more, around 12 genes, but those were not high yielding. And for us, the high yield and the good plant type was necessary. So these lines are now available for testing, and these lines will be now moving to the multi-location trials, and you can see that the how much is the yield advantage of these lines under normal situation as well as under drought. Now the whole exercise that we can develop a drought tolerant variety with the same high yield potential and disease resistance and all the genes together is possible, provided we use marker assisted selection or marker assisted by cross breeding properly. And also, as I said in the beginning, that when I started research on drought, I never dreamed that one day I will think of combining 15 or 16 genes together. Even if I succeeded only 10 genes, because in the F2 we went ahead with only 15,000 plants. If we would have had double the plants of that, we could have succeeded in fact bringing the additional genes also. But having said that, it was not so easy. You have so many complications. You still get linkage drag. You need to have a very good phenotyping. You get many undesirable linkage drag which you don't know, and that you can get rid of only by phenotyping. Wherever you know, you can deselect or you can reject for such markers, no, or you can select for the against markers. But wherever you don't know that these linkage drags are happening, phenotyping will help you. And I think for many of the undesirable linkages, we are still reporting, we don't know. And so we decided that we should have comprehensive phenotyping, and that is why I told you that even if we had plants with 12 genes, we didn't take it forward because we did see that their yield was low, so there was linkage drag. Now these lines can become variety, but these lines will also make people's life easy not to go for the complex process that I had to go if they want to combine the number of genes together. And these lines are available if people want to use in their breeding marker assisted breeding program to develop a comprehensive variety having disease resistance, drought tolerance, insect resistance, submergence tolerance, and move ahead for testing those varieties. So as I was saying that one example was the QTL2 variety, where the drought and submergence tolerance were combined together, 4 varieties developed. But then DBT also gave us an additional project with ED, TNAU, IGKV, IIRR, and PAU, where we worked to improve 12 varieties for diseases, drought tolerance, submergence tolerance, and in fact you can see that people have combined 8 genes, 9 genes, 7 genes in the program, and this is being done by them. So this is the one at PAU they have done it, the one at IRR they have done it, the one at TNAU they have done it. So we will be moving towards the last section now, and as I was saying that as technology improves, the precision in the breeding should improve. And so now we can track markers, and then we can say that this line has this marker and therefore has this resistance or tolerance to this disease or drought based on that, and we say it with a quite confidence. And when this marker assisted technology started, at that point of time we didn't believe that this will happen one day. We thought that this is only for using traits and identifying traits or basic study. But evidently, this is helping breeding, and this is helping to make breeding more precise than what we were doing earlier. So what is needed in breeding? If you see this slide, at least we have illustrated 21 traits that probably a breeder needs. There are some new traits: low GI for diabetic people, high iron, high zinc for nutrition. This I am talking about rice. So many quality traits: low, medium, high amylose, low chalkiness, no tolerance to temperature, heat, cold, submergence, stagnant flooding, drought, anaerobic germination, root plasticity, lodging resistance, number of tillers, blast, BPH, BLB, how many panicles per square meter, grain per panicle, steady stem, and all this. And evidently, for most of these, there are genes or QTLs known. So can we do precision breeding? That that I decide in my mind, like doctors do, that how many centimeter incision they have, millimeter incision they have to have when they want to operate. Can we decide and arrive at that result that I want rice variety with 90 days, but it should yield 6 tons and it should have these traits, and then through precision breeding we make that possible. Will it be possible? Yes, one day it will be possible, and it will become possible only when we start thinking in that direction and we start using the technology. So we will talk that how we move towards that technology. uh, as technology improves, we give you one example with days to flowering, that we are moving towards that. But let's talk about the haplotype. So two discoveries happened in 2017 and 18. I would say discovery, even if it was a report. Nature, which said for genes have different haplotypes. Some of the haplotypes, they are superior than the other haplotypes, and you can see here in the haplotype assembly part that for the flowering time there are loci which is H3, which is behaving differently from the H2 and H1. For the yield, you can see the haplotype H2 is behaving different. For the disease, you can see that the haplotype H1 is behaving superior to other haplotypes. In again, you can see the H1, and Bivan proposed that we can combine these haplotypes, superior haplotypes, using assisted breeding. And this will bring lot of advantage than just gene-based breeding that we are doing. So then the second question or second report was that in the rice, again the Nature review, that Bing et al. Reported about the characterization of 2,200 genes which has been characterized, and which includes genes for yield, growth and development, disease resistance, nutrient efficiency, fertility, insect resistance, grain quality, stress resistance. So there are genes. There are 2,200 genes having different traits that we can use in the precision breeding program. So we initiated a study that whether these genes uh, that are reported, whether they have more than one haplotype. So we started with a smaller subset, and we found that in majority of the cases, 95% of the functionally characterized genes have extensive haplotype variation. And in fact, in some cases, the number of haplotypes that they carry is up to 15. There are very few that they have only one haplotype. And whether this is for the disease resistance, whether this is for yield potential, whether this is for the stress resistance, this is true. So we took around 120 functionally characterized genes, and you see here that the number of haplotypes that they are present for the different genes, in most of the cases except for few, you will see more than one haplotype. that are present. So means within for the different genes, we can use superior haplotypes and we can increase the response or increase the trait value. So we took initially 21 strongly associated genes and which are located on different chromosomes for the yield, tiller number, primary branches per panicle, plant height, panicle length, days to flowering, grain size, amylose content, grain iron, grain zinc concentration. And you can see that the superior haplotypes, they do increase the value. If you see the PBE percent, 19%, 23%, 15%, 9.8%, 10%. So there is a chance that we can increase the effect of these traits by using the superior haplotypes. Now, to our surprise, we wanted to see, once we identified the superior haplotypes for the different genes, for those 21 traits, we wanted to see whether our mega varieties have those traits, have those superior haplotypes, IR64, Swarna, and to our surprise, that IR64 has only one superior haplotype, and which is for the days to flowering, and Swarna has superior haplotypes for two traits. One is for the filler number and one is for the days to flowering, but others are not there. Others are not there. So there is a great chance that we can improve the yield of even these varieties by using this superior haplotype. This is true not only for the convention, the marker breeding, but if we do select those donors which have the superior haplotype for the different genes, it could be true for the conventional breeding, and it could be true for the genomic selection approach, or it could be true for the genome editing if we want to edit the gene. And so in this slide you see we have reported the superior best haplotype for the different genes, and you can see the GSD7, which is flowering duration, we have reported three haplotypes: H8 if you want early duration variety, H14 if you want medium duration variety, and H6 if you want late duration variety. But again, as I said, it's not as easy as we said, but it can be done for sure. But if you want to do the breeding for the superior haplotypes, we have to identify the superior haplotypes. We have identified for some, and we will be reporting for the superior haplotypes for most of the 2,200 genes very soon. And then you have to have a trait development program. If it is coming from the line races, these superior haplotypes have to be brought into the improved plant type so that we can use it into breeding without any linkage drive. And if it is in the improved plant type, then we can use it. For some of the traits for which we don't have, we may have to identify the QTL and the gene and then the superior haplotypes. And as I said in my previous slide, once you are there, you can use conventional breeding strategy, you can use marker assisted backcross breeding, or you can use marker assisted selection, or you can use genomic selection, or you can use genome editing. So this is the story for rice, but this is true for other crops also. This could be true for chickpea, that we have recently seen a paper in the chickpea on the superior haplotypes. This could be true for maize, and this could be true for any crop. So this hypothesis of using the superior haplotypes of the genes could be true for any crop. So how much yield advantage the superior haplotypes that we have reported can increase the number of tillers by 20%, can increase the length of the panicles by 27%, can increase the higher yield by 26%, can increase the iron content in rice by 46%, can increase the zinc content in rice by 34%. But these are the direct values. When you try to improve these traits, there would be compensation, and so when you increase the number of tillers, there would be some compression on the panicle size, or when you increase the number of grains per panicle, there could be some compensation on the grain weight. So that we still don't know, and that we still have to work on. But individually, you can see that we can improve these traits. So we wanted to see, as I told you, that there were three genes which are responsible for early, we can have medium, we can have late. We did a, we carried a simulation, a small simulation for our paper with a training set of 100 lines, and we wanted to test whether we are able to manipulate the flowering. And even for in the early duration lines, by using the different haplotypes, we were able to reduce the flowering and manipulate the flowering by 6 to 8 days with the accuracy of 0.7. So this was a small simulation. So if you increase the simulation size, you increase the accuracy, and you also increase that how much you can control. And thus we have tried to prove that we can move towards the precision breeding for the different traits, and we will be proving for each of these traits in time to come. For the grain yield, where we will say that we want to achieve this grain yield, and this is the superior haplotypes, and then we will prove it through simulation that this is happening, and not only through simulation, but we will do the real breeding, and then we will try to show that how as near we can achieve to that simulation. So as I was saying that that it's very easy to say that use the superior haplotypes and you improve these traits, but like for the QTLs, we will still have the problem of the undesirable traits due to linkage drag. For many of the traits where we have not broken, we have to break the superior haplotypes. There will be some compensation effect of one trait over the other, like increasing panicle length and number of grains per panicle may not follow a linear relationship. And these compensation effect we still have to study. For the drought, for the QTLs, as we moved on, we always used to get new challenges, like we first initially identified the positive interaction between QTLs, but then we also identified the negative interaction between the QTLs, which decreases the yield. But then we identified the interaction between QTL and the genetic background, and then so many these interactions were identified, and so we still have to study more. But I tell you that if you combine your technology used with the phenotyping, proper phenotyping, I think you are able to combine, and you are able to arrive at a win-win situation. So this is the story that I had over last I would say 14 years of research on the drought tolerance that we did. And one idea of giving this presentation was that yes, of course, to present the progress, but I tell you, and to encourage people to use these QTLs and the improved lines if they are breeding for the drought tolerance, if they are taking a program on breeding for the drought tolerance. So they use these lines rather than they use the traditional line. But the larger aim of this presentation is to improve, to encourage young researchers or researcher at any age, I would say, to continue their knowledge gathering ability. Don't leave it. This is a competitive world. When I started with a conventional breeding approach, and in fact, first of the few publications that I have, which was did mentioned during my introduction, the 7 Galvani resistant genes, they were done through the classical genetics. So I came as a conventional breeder, but I always try to learn the technology, and that is where from conventional breeder I tried to become a marker-assisted bed cross breeder, then to marker-assisted selection breeder, then to go towards the genomic selection, and then we are talking about the haplotypes, superior haplotypes. One way that it helps the people who are getting into the last quarter of their career is by engaging with the young PhD and master's students who understand the technology far better than us, but then and they can explain you in a more fantastic way. So the larger idea is that keep yourself moving forward with the technology, with the knowledge, so that you remain competitive in this world of research, which is going to be more and more competitive. So last but not least, thanks to all those donors who have supported us, right from Bill and Melinda Gates Foundation to Department of Biotechnology, to Indian Council of Agriculture Research, to Harvest Plus, to CGIR, to many other donors which may not be here. And I think many of you may be knowing that the Rockefeller Foundation used to be a uh, very large donor in India, and they have trained many people in India. And in fact, I am one of the beneficiaries who is uh, talking in front of you. In fact, they were the one who sent me to Erie for the postdoc. So once again, thanks to organizers for inviting me, for reaching out to me. I don't know from where they got my phone number and the email address, and and how much effort they must have put in to decide that they should invite me. So thank you very much for the whole team uh, to invite me and to get this linkage with the virtual linkage with a set of people who are participants to this program. So thank you very much, and thanks to all the participants for listening to me attentively. I hope that this lecture is beneficial to you. Thank you very much. I'm open to questions.
Thank you, sir. Thanks a lot for your detailed lecture. This was a pure science over here. I can say only that. It was a pure science discussed today. And if I can link this towards our theme, that's a zero hunger. This is the way forward. We can enrich our rice as far as zinc and iron is concerned. So that is the way, the nutritional security we can achieve. The staple food of our rice can be enriched with iron and zinc, and which is needed. And we in our population, we have the deficiencies of these micro minerals. And the other way, this is the way where we can produce resistant or you can say the tolerant varieties of the rice to drought or to floods or to other these infections, or you can say the other diseases. So this was a nice lecture, and a way forward to achieve such goals in achieving the zero hunger, our theme of this training program. So this is the way the biotechnology can work there, increasing the yield, increasing the varieties with micro minerals, increasing the varieties which are resistant to the diseases and other climatic, or you can say the abiotic stresses. A nice talk by Professor Arvind Kumar, and really he has done a lot of work in this area, the rice, and I think I must salute you, and nation must salute you for your such endeavors where you wish to have the varieties of staple food, I can say the rice, in our nation or throughout the world with such qualities, with such QTLs and all that. So I am from basically from animal science. I think doing such things in the animal sciences, it's okay there, but it's not so fast there. It's a very slow process in our animal science. And we are fortunate enough that this time on one side of this screen, I have a biotechnologist, plant biotechnologist on. At the same time, Just on your left side, we have a renowned animal biotechnologist of India, Professor ML Madhan with us. He is there with us in this meeting also there. So I welcome you, sir, Professor Madhan, sir. If you can unmute yourself, please. You can unmute yourself, please. Yes.
Pleasure, Dr. Pampuri,
Most
and
my pleasure. Sir,
Arvind enjoyed your talk.
yes, yes. That's
Thank you, sir. Thank you very much, and good to get connected with you, even if virtually. Thank you. Very good to see you, sir.
that's
Yeah,
we are blessed this morning that we have two biotechnologists, one from the plant science and other from the animal science, and both are the reputed nationally as well as internationally. So most welcome to both the speakers here. Sir, if we have any questions from the participants, please raise your hands. Is there anyone who can who wish to have an interaction with our speaker? I think mostly they must be from the agriculture sciences. Anyone from the agriculture science, particularly from a breeding side? We do have so many there in. I mean, the participants from the breeding is.
personally, Dr. Pampuri, I see this as a true future for the country in terms of the grain yields and in terms of the
That
food requirements of our country.
that I saluted him. I said that nation should salute such a personality. They are working in such an area where you can have biofortification as well as the yield.
And as you said it, that we are a little envious because of the fact that a lot of progress can be achieved over into the plant side over year,
Exactly.
with the generation intervals being so small. But it has been of late that in livestock also that tremendous progress has been made.
Yes.
Great strides
Yes.
have been made, both in
Yeah.
small ruminants and large ruminants. And that's what sustains us. As a matter of fact, the sustenance in the country
Yeah.
today, in terms of its nutritional sustainability and food sustainability, is stemmed to the livestock. And we have a lot of materials going over in the laboratory. But it's very difficult to make them travel over into the farmers. Primarily for two reasons. Farmers are untouched with their own thoughts, and they're so scrupulous about their own varieties.
Yeah.
And you try to introduce with them unless and they are convinced that yes, they have something, they will not accept it. And that's one real bottleneck. And the second thing is that we have till now not cultivated an habit of rewarding and grower, a farmer, to the quality he is producing.
Yeah.
So he is not bothered at all at the quality. And therefore, that when we talk of quality crops, when we talk of abiotic stress-resistant crops, when we talk that they can better have the resilience to climate, the farmer does not feel interested in that because we have never given him an opportunity that we should reward him for his quality. I have all the time been pleading for this fact that when we are fixing the price, let's not bind all the produce into one gap and into one slot. Let us see that give prices according to the quality which is produced. Whether there is about 18% moisture or there is 11% moisture, the farmer gets the same thing. So he's interested in having more moisture into it. If it's not natural, he will put in moisture into it so he gets a better weight.
Yeah.
But if you have gotten quality, whether you have gotten quality wheat, new varieties of quality, new varieties of rice and all that, we do not go our way out to pay him for that quality. And I think in order to, Arvind, in order to see that your new varieties, where new transplants are getting better receptivity, let's put together these hands and see that the farmer gets better rewarded. Thank you.
True, sir. This is true. You are true. So what is next? So what is next step for us?
Firdaus, can you unmute yourself, Firdaus? Hello, Firdaus. Firdaus, can you unmute yourself, please?
Yes, sir. Assalamu alaikum.
Walaikum salam. Can you have an interaction with our speaker over here?
Yes, sir. Yes, sir. I can, sir. Sir, a very, very nice presentation and insightful one, sir. I feel like since we are dealing with genome-wide information and QTLs are large enough so that they need first the basic data that we are generating, second, the bioinformatics analysis. Sir, besides ISRI from New Delhi, is there any other institute that is accessible to students so that they can learn different data processing softwares, data processing techniques? And then if we can move from now we are moving from QTLs to SNPs. Like we have bead chips that can produce a huge number of SNP marker data, maybe 50,000, maybe in lakhs also. So we need that bioinformatics support at national level and at state level also for faculty also and for students also, so that the manpower and skill power is enhanced at that level. Sir, I want to ask whether besides ISRA at New Delhi, is there any other institute that is providing that access to students as well as faculty?
Yeah, I think this is a good question. You know, DBT has started developing a platform at NIPGR. If you have heard about the NIPGR,
Yes, yes.
and you know, and this should be available to the researchers. I would say next 2 months. In fact, they have hired some people working in the private sector to quickly establish this platform. So while ISRI is there, the NIPGR will also have that platform which people can use it. So, and more facilities will come. In fact, ERI is also trying to develop a data management system at our center in Varanasi, and DBT has provided us some funding, good funding support, which we believe should be available in these 6 months, and probably many more will come. So the system has realized that people need support on data management and bioinformatics, and facilities are coming up.
Thank you, sir. So I want to ask one more thing. Sir, mostly we go for outsourcing for data generation regarding QTLs as well as SNPs. Sir, what is Government of India and scientific fraternity doing so that we do not outsource to exotic foreign companies? We outsource only to maybe indigenous or that are producing, that are generating the data within India.
So evidently, you know, most of these companies, except for BGI, most of these companies, they have their labs in India. So you don't have to go to outside. That is one part. The second part is that even for the outsourcing, again, the DBT is initiating a facility where you can get your the genotyping done in the national system, and you can get your genotyping done even at the current what is that facility, NIPB, at within the IRI campus. Even they are supposed to do, and NKSing has generated the SNP panels for that. So there are different that you can select. And I think, yes, we have moved into the version of the outsourcing of the genotyping, because it has been very difficult for the institutes to move forward with the way the technology change and put in the money to bring the new instruments. And so the public sector thought that, you know, let's move towards the outsourcing, and that's not a bad idea. But then public sector are also developing those facilities. The national system is developing those facilities, which will keep on moving. So this will happen.
Sir, one other opinion I have, sir. It's not that I'm making that opinion randomly. Sir, I feel like we should make it mandatory that after an embargo period, after the generation of data from a project, it should be made available to public so that we can work upon that. Or if we are generating new data, we can club the previous and current data, and then we will save resources. Our publications will be fine, and our animal number or variety number of fields will be enhanced. So that will help overall in research building.
Yeah, means as an institute, ERI believes in open sharing of the data, and so if you remember, we sequenced 3,000 germ plasm, and we have provided open access to data to all researchers, the sequencing data. Very rare to happen, and we have done that. So the national system has to decide, initially that they provide access. First thing is that we have access at somewhere where it is stored for the national interest or the university interest or the provincial interest. And then once the researcher who are associated with generating the data, they have used the data for publication and everything, those kind of data should be made accessible to everyone, and that will in fact help science rather than limit science. So as an institute to which I work, we believe in open sharing of the data, and in fact the day we had 3,000 rice genome sequenced, ERI provided open access to that sequencing data to everybody. So even you can go and you can access the data. And that was also done with the idea that probably many of these public institutes will come forward like this and will promote open sharing of the data. But it should happen, definitely it should happen. And one day it will happen. At least it should happen after the concerned researcher had published the part of the research or the research that he or she wanted to undertake.
Thank you, sir. Thanks a lot. I have a question from Ashwini Kumar in my chat box. He says, is something going on at national level for mushk budji and red rice of Kashmir?
I'm not aware of. I'm sorry, but if you feel that you must be proud of the red rice in Kashmir. So why not we initiate from Kashmir? Let's have a project from Kashmir funded by DBT, and you can engage with one or two partners from outside whom you think have that technology that you want to improve. I am not aware of anything happening like that, but why not your institute start that, and let's do that.
Yes, they are there. Our university, that's Sukhas Kashmir.
Yeah.
That's doing a good work towards this mushk budji as well as red rice. Probably they are reviving all those varieties now, the local varieties, the indigenous varieties. Yes, there is good work going on in those two lines, the red rice and mushk budji. And even in case of the apples like Amri and all that, what we had lost, now they are reviving all those varieties now. That's there. So if we don't have any more questions, may I ask this Hamdani, Hina Hamdani, are you here?
Yes, sir.
Am I audible to you?
Yes, sir.
Can you just say thanks to our today's speaker before we end this meeting?
Sure, sir. Good morning, everyone. Listening to an eminent scientist of such great repute is indeed a privilege for all of us. Sir, you have truly inspired us all by your great work. We are really grateful to you this morning. This has been an informative and elucidative session. I hope all the participants have benefited from this lecture. I thank you on behalf of our university, the organizing committee, and all the participants for sharing your knowledge with us. Thank you very much, sir, and have a very good day. Over to you, Pampuri sir.
Thank you, Dr. Arvind Kumar, sir. We are really blessed to have you this morning, and it's raining in Kashmir like anything. So outside, I have too much noise. I don't know what is the condition in Delhi itself.
No, I'm at Hyderabad as of now. It's not raining here.
Okay, okay, okay. It's heavily raining over here in Kashmir. Probably for, I think after a very long dry spell and high heat, now it's the day. Today we are enjoying the rains over in Kashmir, and it has dropped the day temperature also. So we are very thankful to you, sir. Enjoy and Hyderabad, your stay, and then be safe and be blessed, please. And we look forward for your cooperation in future too, sir.
Yeah, my pleasure. And thank you very much for inviting, and good virtually meeting you, Dr. Madan. Thank you very much.
My pleasure, sir.
Yeah. Bye for now. Thanks, everybody.
Thank you,
Bye.
sir. So before we leave, we have with us one participant, that's Sheikh Firdaus. As already discussed, he is one that he will present his presentation, his views, and just his ideas about this theme in a very spell of 15 minutes. So may I invite Firdaus? Can you unmute yourself, Firdaus?
Yes, sir.
Yes, you can proceed with your presentation, but be brief, please.
Yes, sir.
Okay.
So I will leave. Thank you very much.
So nice of you, sir.
Yeah, thank you.
Very good morning to all of you. So, I'm blessed to have this opportunity in presence of Professor and Dr. Amal Madan, sir.
You can just on your video also. You can put on your
Am I
video.
audible, sir? Hello. Am I audible, sir?
Background noise there.
Sir, I apologize. I could have been at home, but since there's 2G connectivity, it will disturb that way. I'm sorry for that. I will be a little louder, sir. I'm blessed to have Professor Amal Madan here with me, and it's been a blessing and pleasure to present my presentation on this. I'm sorry for the background noise at first. So my concept here is, my topic here is targeting zero hunger through innovative solutions of science and technology. I'm Dr. Sheikh Firdaus Ahmed, and I'm a scientist in the Indian Council of Agriculture Research. So before proceeding with anything, we should know the present status of Indian agriculture. We have so many feed crops being produced. If you can see from horticulture crops, from climate resilient varieties, the scenario is very good. These are the national figures and latest figures. If we see the production, we have 2019-20 data with 291 million tons of food grains being produced in India. We have 187 million metric tons of milk, 103 billion egg, and 13 million tons of fish. So production is not a problem at present, at least. We are exporting Pusa Basmati to the tune of 19,000 crores. And we have a sugarcane variety we're exporting it to the tune of 7,000 crores. Then we also have Kuposhan Mukt Bharat programs and other programs also. All we are standing at a very good position. But there's one other side of the coin. We say that Chirag Tale Andhera. I was astonished to know that 1/3 of Asian population, 1/3 of world malnourished and undernourished people are residing in Asia, and India forms a major part. 40% of food that is produced in Indian nation is wasted, and all in all, we have enough produce, but the distribution is flawed. That we need to counter. So my views are, I have named it as step up, step down approach. So in step up, it's mainly concerned with farmers. In step down is mainly concerned with government officials. We need to improve the production. We need to enhance the productivity. We need to enhance the land holding. Land holding and animal holding is a big challenge to Indian agriculture. How we will improve the land holding, I will come to that. We have to equalize the distribution. Again, I will come to that. And support to farmers has to be increased. Official penetration and reach to land from lab has to be increased. Adoption and acceptance of technologies, cultivars, varieties has to be enhanced. And we need to revisit our strategies so that we are having a very good position in future also. So overall, innovation is the key here to reach zero hunger. Uh, so innovation needs transforming agrarian economy, transforming agriculture of India, and it needs innovative solutions through agriculture and science and technology. So in India, we had maybe 3 or 4 decades back, we had more than 50% of GDP being contributed by agriculture, but presently it is only 17 to 18%. So it's not the flaw of agriculture that its contribution to national GDP is decreased. It is the contribution of other sectors, service sectors, and other secondary and tertiary sectors. Their contribution has increased, which has led to a decrease of contribution of agriculture to national GDP. Before going for any innovation, any transformation, we need to go for SWOT analysis: strengths, weakness, opportunities, and threats. So this I have made a table giving the strengths, weaknesses, opportunities, and threats. We need to think, we need to revisit our strategy so that these weaknesses are converted into opportunities, and we are able to face these threats head on and get triumphed all over. Overall, as I said, innovations are the key, but I want to tell it at a place that there's no one-shot treatment. You cannot say that this one technology is there which will transform each and everything for our good, and so that we are reaching that target of zero hunger. So I am proposing few technologies here, few innovations. All the participants, all the worthy participants may have their own technologies, their own bills to transform the agriculture. So artificial intelligence, IoT, robotics, A&M, image prediction, algorithms, these all have a very, very high role in improving the agrarian economy in India. We have this robotics, fruit picker and crop harvester we can use. So A&M and artificial intelligence play a big role both in agriculture as well as its allied sectors, in livestock also. So we have precision farming. This has been dealt with in detail by so many worthy speakers. This will lead us to, we can use GPS, we can use sensor technologies, we can use GIS, we can use micronutrient and nanonutrient approach. So overall, it will lead us to resource use efficiency so that minimum resources are used, minimum waste is there, our agriculture and our environment will be sustainable. We have another concept of ionomics in agriculture and allied sectors. If we go for ionomics so that we are able to see what is there in animal or agriculture field regarding micronutrients, regarding other elements. So accordingly, we can go for interventions, and we will have proper understanding of gene function. We will go for gene network analysis. We can have better disease diagnosis. We can have disease therapeutics as well. Next thing is thermal imaging and thermography. It has wide applications both in agriculture, livestock, fisheries, and others. We can use thermal sensors for thermography, thermotherapy, and thermal sensors can be used for all these purposes. What are the potential uses of this thermal imaging and thermography? We can monitor the moisture content. We can go for better soil health and management based on the data that is generated. We can go for nursery and greenhouse monitoring. We can make informed predictions vis-a-vis disease incidence and maturity, subsequent shelf life, and ultimate use of agriculture produce. Then, as we had a speaker in the morning regarding QTLs and genomics, so we need to go for this intensive genomics. Big data analysis, we should have that data first, then we should have the infrastructure to process that data and get networks, pathways, and biological things that can be beneficial to us. We need to integrate the omics approach. It's not that we go for only QTLs and genomics, the phenomics and dynamics, or other transcriptomics, metabolomics. We should not go for that. We should integrate all these things so that meaningful interpretations and those results are there which can be applied in the field. We will have with intensive genomics better understanding of traits and diseases. We have to prioritize biodiversity and adaptation. We can establish gene banks and data banks so that gene banks are helpful in conservation and preservation of needs, and data banks can be used for future genetic progress in different breeds, in different varieties of agriculture. These biopeptides and superabsorbent polymers have been in use in foreign countries, and now in India they are being used. I want to quote an example. In mastitis, we are using biopeptides so that it helps in diagnosis as well as treatment. I'm suggesting there's one superabsorbent polymer which can, if it is raining as is presently in Jammu and Kashmir, the polymer absorbs the moisture, and if it is sunny outside, the polymer will release that water. So this can help us in those areas where irrigation is minimal or irrigation is nil. We had I had a reference of Cardozo and co-workers. They in 2014 said that there's a polymer which can release nitric oxide and which can be used for combating mastitis causing pathogens. Then post-harvest losses are a big problem in India, and we have post-harvest losses to the tune of at least 1 lakh crore. So we can go for cold appeal signs. We can use that appeal so that that chemicals are used so that shelf life of a particular product is enhanced. We can use prediction softwares like AI, ANN, and IoT, so that if some horticulture produce or vegetable produce is produced in J&K, we should know first what is the shelf life, whether it can reach to a distant location, for example, the southern states or foreign countries, or it's not optimum, it should be used locally or within the nation, within the state of vicinity. We have a CRISPR system which can be used for quantitative traits, which needs to be used for quantitative traits. These are molecular scissors. In this table, I'm showing a few instances of genes that have been mutated or modified through CRISPR technique, so that there has been enhanced and significant increase in seed size, number of crops, and muscle mass. We have in the livestock, we have a famous example of myostatin gene. Then I want to propose there are 74 million milk cattle as per the census of 2020. If we are able to improve only 100 ml increased milk per cow, if we multiply this 74.18 into 100, then it's a huge figure. Similarly, we have 851 million poultry. If there is an improvement of 1 egg per hen, then it's a big thing that can be there. Then improved greenhouse technology. We have harsh winters here. We can use those kind of greenhouses where heat is conserved, heat is maintained there within the greenhouse in those harsh winters. We can use the geothermal heat, and within those greenhouses, we can use conventional farming, we can use multi-tier farming, we can use hydroponics, we can use aeroponics. So these are all possible if we are dedicated towards research first and then applying these in farms. Then we have farm management softwares. I will not go in detail. Solar power in agriculture. This can be used for desalination of seawater, for irrigation use. We can use fertilizer deep placement. Once it's deep placed, we can improve produce from same resources, whatever we're using. There will be no residues in the environment, and environment can be sustainable. Then we have zero liquid discharge. We are using water for maybe for irrigation purpose, maybe in fisheries, maybe in some other purposes. But we are adding agrochemicals, we are adding other chemicals. Then that water is contaminated. But before disposal and discharge back into the water body, we should process these water resources so that solids are recovered and disposal is free of those chemicals whatever we have used. Then we should also try to go for biological pest control that will lead to lesser reduced use of agrochemicals, so that the burden on environment and sustainability of environment is there. I want to stress on this thing. So I think there's a population dividend in India, that more of the population is in young generation. So young generation is more or less addicted to social media. We use Facebook, we use WhatsApp, we use so many other apps. So why can't the farmer use app? Why can't we make use of those social media apps to reach out to the farmers? We can have a Google chat system which says that, dear farmer, tell me what you want. I'm your like whatever you want, I can give it to you. Maybe in the morning, maybe 24/7 into 365, I'm available with you. So we should make use of all these things. And based on that, we can go for efficient genome recording. We have a big problem of genome recording and reliability here. So once that technology is used, we can ensure proper genome recording. So at last, I want to discuss or give my opinion about 5 topmost innovative solutions that, in my view, are relevant in present conditions. As I said at the start, that the participants, the worthy participants, can have their own view. They have the right to agree or disagree or have their own opinion about this. I am proposing farmers markets. This should be a routine activity, so that whatever farmer produces, there's a market where he or she can sell. She can sell the produce, and it's not rotten for the cause of maybe transport, maybe infrastructure. So if those markets are accessible, it will help the cause of farmers. Then we have to assess the agricultural produce for its shelf life. For example, we have an apple, which we need to see its ingredients, its sugar content, its carbon dioxide content, it's all the things, maybe microminerals, maybe microelements, maybe some of the things. We need to assess those things and say that its shelf life is only maybe 7 days, so it has to be consumed locally. And if its shelf life maybe 3 months, 4 months, it can be sent outside states and outside countries. This is possible with the artificial intelligence and the artificial neural network. We need farmers dedicated teams that are with the state-of-the-art technologies, having that micro or macro atmosphere, that farmers' yield is not rotten, farmers' yield is maintained within its conditions, so that farmers' yield reach the target, reach the market without much damage. Then we have a large fake agrochemicals. This is rampant in India, and Indian farmers are at loss because of this. So I am proposing one thing. Maybe a barcode can be used that is published or printed inside the bag of that fertilizer, or a number is printed on that inside the bag of the fertilizer. Once the bag is opened, we can send that OTP, that barcode or a number to some phone number, so that it confirms the authenticity of that agrochemical. For example, there's a agrochemical which is passed, and its cost, for example, its cost of Kashmir says that it is passed, its quality is standardized. So we have to use innovative solutions so that yes, farmer is ensured that yes, his chemical or his seed or his pesticide is having the appropriate quality for his seed. So how can we improve the land holding? As I said in the start, Karnataka government and Himachal government, they have used these helicopters and drones so that seeds are thrown out into the barren lands, and if we are able to convert a small piece of land at each district or at each village, it will largely solve our purpose. Thank you so much for taking the time and opportunity.
Thank you. Thanks a lot, Sheikh, for those very nice presentation. I can say this was altogether concise. Then you can say the roadmap actually for us, for all the scientists, for in future to see that our country or the whole globe is with zero hunger. This is really a very nice presentation. I say I feel that many of these technologies are there in place, but in developed countries, you know, when we talk of zero hunger, all these things are there. But yesterday we had a speech from our speaker, and then I think that was a very nice point he made. It's zero hunger cannot be achieved altogether by only targeting a single goal, zero hunger itself. No, it has to be a collective. Say you have to look into the poverty, you have to look into the health, you have to look into many other, those sustainable tools. Then only you can have a zero hunger. So if you have a poverty, you have a produce is there. Okay, as you said, this produce enough even to feed more than 9 billion people. But the thing is, accessibility is not there. Purchasing capacity is not with the people. That's the problem. So you cannot think, in isolation, achieving zero hunger only by these means. You have to see other things there. and the technologies you are talking of. Again, we have discussed that probably in a whole of our, those previous days, that this land holding is so little with Indian scenario, that your technologies are they are not I can say they are not adaptable there. They cannot adopt those technologies in our small land holdings, and for that we need the technologies that can reach to a farmer, a small farmer, and he can utilize. You cannot have a thrasher, a big thrasher, who can go into the hectares, 20, 100 hectares of the land. We have a small 1 hectare land, and you need to have some small thrashers. You need to have some small choppers. You need to have something which is really applicable in our land holding situations, in our that conditions, and at the same time in our economic status. We need to have those technologies which are feasible, which are you can say accessible, and that's why our those I mean the Western countries, they are economically very sound, and they do utilize those technologies. You have those you have seen in your those I mean slides, automatic that seeding is there through robots, what you say, name those. I am not getting that term in my mind. So technologies are there, and in future we look that in future this is the way only. I can say this is the only way in future that we will have to adapt those technologies. We will have to go digital. There's no other way. Even at this time there was no other way to go digital because of this pandemic. Otherwise this time we must be there in our I mean labs or in our labs. So probably time is coming. Time will compel us. I think time will compel us to adopt those technologies, and it's nice to hear you here. Even I'm getting in my chat box many messages from participants. A very nice presentation. And if in the audience, in the participants, if anyone is there who want to share his views or who want to have some interaction with our speaker, Sheikh Firdaus, you can please raise hand. There any who can just have an interaction with our Firdaus? If no one, then it's okay. Hina want to say something. Hina, you can unmute yourself. Hina, yes, you can unmute yourself.
Good afternoon, everyone.
Very good.
Dr. A very good presentation. You have shown us really roadmap what we should do in future. Well done. Keep it up.
Yes, yes. We can only pray over here for his future, bright future, and we believe in him. We know him. He has been our student and bright student. That's there. Professor Sir Madan sir.
उनके दीदार तो कराइए. I don't see his face.
वो मैंने पहले भी कहा था कि पर दीदार दे जिसे नहीं दे रहा है, पता नहीं उसका भी. हाँ, आप दे रहा है सर, आप दे रहा है सर, आप देख रहे हैं सर उसको.
Firdaus, you have done a good job.
Thank you so much, sir.
That's I would say. बड़ा neatly, cleanly आपने presentation बनाई है. Though this thing is being said over and over again, yeah, and that has to be said, because the more we say about it, the more it percolates down.
Yes, sir. Yes.
But these are the things on surface. I hope you understand that.
Yes,
But I am admiring your the NACA presentation and the material you put together for this presentation. अगर कुछ नहीं तो आपके हम जमातियों से ये मेरी इल्तिजा है कि वो देख लें कि presentation कैसे होनी चाहिए.
Yes.
Irrespective of the material.
Thank you so much, sir.
मैं देखना चाहता था, आपका चेहरा देखना चाहता था.
So we have met, sir. I think I remember one thing of yours.
Thank you, Sheikh.
Yes, sir. Yes, sir.
He has been there in our SAPI also. He has won so many medals in SAPI quiz also, sir. Is it so?
Good.
Firdaus?
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
Yes, sir. He
He has to do it.
happy,
He has to do it. See, I can see him with a very clear focus.
Okay, okay, sir.
Thank you so much, sir.
So, Hina, can you just pay thanks to our speakers, to our Professor Madan, so that we can end the meeting? Hina, are you there?
Yes, sir.
Please.
Professor Dan Saab and other dignitaries, we are really grateful to have all of you with us this afternoon. It has been a pleasure for all of us. I thank you all from the deep core of my heart on behalf of our organizing committee, our university, and all the participants who are listening to you. I'm very thankful to Dr. Firdaus as well for a very good, nice presentation. I wish all of you good health, stay safe, stay blessed, and have a nice day ahead. And those people who are in Kashmir, enjoy the rainfall.
Yes, yes, yes. That's the, after a long spell, we are now enjoying some relief in temperature, day temperature. So thank you. Thanks a lot to everyone, and particularly my Professor Madan, sir. We take leave over here and we'll be assembling at 2:30. We'll have one more speaker at 2:30. Till then, stay safe and stay blessed. Thank you, sir.